Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry mentor. Now in this video we are going to discuss about the synthesis of fatty acids, which is known as lipogenesis. So let's start with the discussion, because in my previous videos I have made one point clear to all of you that whatever anabolic processes are there, they will always take place in the cytosol, and we generally put a suffix as genesis with them. Okay, so now lipogenesis. So it means that genesis means formation, lipo means lipid. So simply lipogenesis is formation of lipid, and I have told you that lipids we call them as a clinical terminology for lipids is TGs. So we can say that synthesis of the triglycerides is basically lipogenesis. Now the question is, it is an anabolic process. And we all know that anabolic processes always occurs in the cytoplasm. So always remember that this lipogenesis takes place in the cytoplasm. Then the third point to remember about them is in what stage it occurs. So always remember, anabolic means the body is synthesizing something. So it will occur in the well-fed state. Right? So site is the cytoplasm. And obviously, where it will occur? They occur, or you can say extra mitochondrial, whatever you can say. So they will occur in the liver, it will occur in the adipose tissue, in the mammary glands, and so on, wherever fats are required. Clear? Then coming over to this, the most important one is, we call this lipogenesis as de novo. Now, why de novo? Because from a smaller molecule, we are synthesizing a bigger molecule. Okay, so the precursor of fatty acids is acetyl-coenzyme A. And how many carbon atoms are present in acetyl-coenzyme A? 2 carbon atoms. So this acetyl-coenzyme A is required for the synthesis, because in our body mainly palmitic acid is produced, right? So palmitic acid has 16 carbon atoms. So how from a 2 carbon species we are forming a 16 carbon species? So this type of process is known as de novo. And it is also known as line in spiral method, because it was discovered by that scientist. We can refer it as lipogenesis, plain and simple, right? Now coming over to this, we have discussed the site, we have discussed how it is occurring. Now the question is, this acetyl-coenzyme A, it is present in the mitochondria. Why? Because the oxidation of fatty acids is taking place in the mitochondria. If we have to synthesize a fat, this acetyl-coenzyme A needs to be transported from the mitochondria to the cytoplasm. Okay, again I will repeat this, that always remember, formation or anabolic processes takes place in the cytoplasm, and the catabolic reactions in general takes place in the mitochondria. So here the same case is there. The breakdown of fatty acids takes place in the mitochondria, but synthesis of fats has to be taken place in the cytoplasm. The precursor means from where it is synthesized, that is already available in the mitochondria, so it has to be transported. Okay, so whenever we talk about the steps of lipogenesis, the first step I would say would be transport of the precursor or transport of acetyl-coenzyme A from the mitochondria to the cytoplasm. Now see, The tragedy with acetyl coenzyme A is it is impermeable. This acetyl coenzyme A is impermeable to the mitochondrial membrane. So what happens here is here a advanced shuttle system is operative. So what happens here is we know, say this is the mitochondria and this is the cytoplasm. So what will happen here is that this acetyl coenzyme A, which is coming from the degradation of fatty acids, right? This is combining with oxaloacetic acid, which is normal TCA cycle, forming citrate. And the TCA cycle is going on like this, 𝗵𝗵𝗵𝗺. Now this acetyl coenzyme A is impermeable. It cannot enter directly into the cytoplasm. So what happens is this citrate or citric acid will be transported. And this citrate will split up into from where it is produced. In the presence of citrate lyase enzyme which requires ATP for the splitting. So the enzyme here is ATP citrate lyase, and this citrate is broken into acetyl coenzyme A and oxaloacetic acid. Okay, and this is known as citrate shuttle. So you can see that there are two types of shuttles which are operative here. One is whenever the fatty acid has to undergo degradation or breakdown or oxidation, it has to be transported from the cytoplasm to the mitochondria via carnitine shuttle. And whenever there is synthesis of a fat, then it has to be transported, the precursor has to be transported from the mitochondria to the cytoplasm via citrate shuttle. So two types of shuttle systems are there in the metabolism of these fatty acids, right? So we can say that cytoplasm mein this acetyl coenzyme A comes. That's it. Now it can undergo the synthesis of fatty acid. Then coming over to the second step. Once it is transported, second step, which is the most important step here, carboxylation of acetyl coenzyme A. Carboxylation of acetyl-CoA. Now see, what do you mean by carboxylation? Now we have to increase the number of carbon atoms because we are synthesizing fatty acid, isn't it? So carboxylation is simple addition of one carbon. And always remember, in general, whenever we talk about carboxylation, the enzyme has to be carboxylase, right? And the coenzyme is biotin. Biotin, which is vitamin B7. And there is a mnemonic to remember this, ABC, where A stands for ATP. ATP will always be required wherever biotin is carboxylating. B stands for biotin and HC stands for this bicarbonate ions. So by the effect of this, we can say that carboxylation means 2 carbon atoms of acetyl coenzyme A will be converted into 3 carbon atoms. Now since carboxylation of acetyl coenzyme A is taking place, so the enzyme would be acetyl coenzyme A carboxylase. And the reaction would be acetyl coenzyme A having 2 carbon atoms is converted into malonyl-CoA which has 3 carbon atoms. And the enzyme acetyl-CoA carboxylase requiring ATP, requiring B7 and requiring bicarbonate ions. And that is why this is the rate limiting step of the entire. So acetyl coenzyme A undergoes carboxylation in the presence of enzyme acetyl coenzyme A carboxylase forming malonyl coenzyme A, which has 3 carbon atoms, right? And this becomes the rate limiting enzyme also, because it is limiting the flux of malonyl-CoA. Because when we are going to study the regulation, then you will actually come to know what is the role of this malonyl-CoA there. So it becomes a rate limiting step of lipogenesis, rate limiting enzyme, right? So what is the rate limiting enzyme? It is acetyl coenzyme A carboxylase. Now we have acetyl coenzyme A, we have malonyl-CoA. Then what is the third step? Third step is the last step. And here the entire responsibility is taken over by fatty acid synthase complex, which is a very, very important example of multi-enzyme complex. So, fatty acid synthase complex, or it is also known as FAS. We can call it as FAS also. So, it is a multi-enzyme complex. Now, what are multi-enzyme complexes? We have already discussed in our previous videos like PDH pyruvate dehydrogenase complex, which cannot work without the coenzymes and the enzymes. So, we know if we see the structure, PDH has B1, B2, B3, B5 lipoic acid as the coenzymes and even E2, E3 as the enzymes. It is multi-enzyme. Similarly, alpha-ketoglutarate dehydrogenase, and a very, very important enzyme of TCA cycle. Again, it is an example of multi-enzyme complex. Similarly, the next one is fatty acid synthase complex, which helps in the synthesis of fatty acid. And always remember, it works like a frisbee, like I would say saucepan. So, it has this type of structure like this, and internally it has 2 active sites like this. This is ACP, and ACP stands for acyl carrier protein. Acyl carrier protein. So, you can see that 2 things are projecting. So, this is cysteine SH. It is having cysteine at one of the active sites, and second active site has pan SH. So, these 2 are the active sites. Okay. Now see, what is happening is this fatty acid synthase complex consists of 6 main enzymes. So, it is like 1, 2, 3, 4, 5 and 6. So, what are these subunits? Basically, what is the reaction here? Because ultimately what is happening is that this is the one which will lead to the formation of fatty acid. Just say if we talk about palmitic acid, we know that palmitic acid has 16 carbon atoms, and we have to synthesize a 16 carbon species from a 2 carbon species. So, obviously it has to undergo many cycles. Okay. So, if we talk about the structure of this, it has 3 different units, right? One is condensing unit, which will condense leading to reduction, reducing unit, and the third one will be releasing unit. Once the product is produced, it will be released. So there are 3 units attached to this. Right. Now what happens here is this condensing unit will have MAT enzyme, okay, which stands for malonyl-acetyl-CoA. And then the second enzyme is, this is first enzyme, and the second enzyme is keto-acyl synthase. Okay, then reducing unit mein there will be obviously a reduction. So it will be keto-acyl reductase. First of all, reduction will occur, which will require NADPH here, not NADH here, NADPH will be there. Then enoyl reductase, enoyl will be produced, and then dehydratase. And then releasing unit is there. There thiolytic cleavage was there, here thioesterase will work. Thio group will always be involved here. Right. So this is the structure of fatty acid synthase complex. So first of all, let's start from the beginning. First is the precursor of fats is acetyl coenzyme A. This acetyl coenzyme A is produced in the mitochondria by beta oxidation. So what will happen is, since it is the precursor to synthesize a fatty acid, this acetyl coenzyme A needs to be transported from the mitochondria to the cytoplasm. But the problem with this is it is impermeable to that mitochondrial membrane. So what will happen is, normal reaction will occur. Instead of acetyl coenzyme A, citrate will come. And this citrate will simply go outside, it will split up, leading to the formation of acetyl coenzyme A and oxaloacetic acid, okay, in the presence of ATP citrate lyase. Then this acetyl coenzyme A, which has already reached the cytoplasm, it will undergo carboxylation. Carboxylation mein it is going to increase the number of carbon atoms. Carboxylation is over, then the third step now will be the task will be taken over by this fatty acid synthase complex. Now if you see the structure here, you can see that it has 2 active sites, cysteine and pan. So what happens here is, this cysteine is always going to accept acetyl coenzyme A and pan SH is going to accept malonyl-CoA. Okay, so what happens here is, this is the structure like this. So if we talk about cysteine, cysteine SH will accept acetyl-CoA and pan SH will accept malonyl-CoA. So this is acetyl-CoA and this is malonyl-CoA. So I can say it has 2 carbon atoms and it has 3 carbon atoms, right? Now rest everything would be the same. Abhi these units will remain like this, 1, 2, 3, 4, 5 and 6. Now what will happen is, this acetyl coenzyme A will be transferred to malonyl-CoA. Okay, that is why the first enzyme is MAT. So we can say that it will be like this. Cysteine SH say this 2, or I will just write 2 carbon atoms here and here 3 carbon atoms. So what happens is, this 2 carbon atom will be shifted here. That is why the MAT enzyme is there. The name of the enzyme is malonyl-acetyl-CoA, because acetyl coenzyme A will be transferred here. And then what will happen is, after this there will be release of carbon dioxide, because one carbon has to be released. So what we ultimately get is this cysteine SH becomes free now. And this pan SH has how many carbon atoms? One carbon atom comes out, 2 plus 3, 5 minus 1, it's 4. So it has 4 carbon atoms. Right. Now this is also known as 𝗏𝗏. Okay. So what is happening is all the units will continuously be released. So first is, you can say that cysteine SH is accepting acetyl coenzyme A, pan SH is accepting malonyl coenzyme A, then acetyl coenzyme A has to be transferred to malonyl CoA or to pan SH so that cysteine SH is free. Okay, so what we get is, the name of the enzyme is, which comes under condense, because condensation is there and there is release of one molecule of carbon dioxide. So we are left with only 4 carbon atoms. Now somewhat reversal of beta oxidation is there. If you see the enzymes, you can see that this is done, then comes reductase. Whenever we talk about reductase, we need to know about NADPH, because here you can say NADPH will be used for reduction, and here it will be a reductase, or we can say simple ketoacyl reductase will be the enzyme. Right. Then again reductase will come and dehydratase means water will come out. Okay. Then finally what we get is, whatever reaction is occurring, this is occurring on the pan SH group, and cysteine SH is still free. So after these 6 activities, what will happen is, this 4 carbon atom will undergo, because we have already increased the number of carbon atoms, it will remain like this. Okay, and then last will be, this will be shifted here. Okay, and pan SH is now free. This is very important. When it is produced, then you can say entire moiety cysteine will be having this ketoenoyl group, right? And this pan SH will be free. And now this pan SH will accept the fresh malonyl-CoA. And again same cycle. Same cycle will go on, because here now 4 carbon atoms are there. Malonyl-CoA has 3. So 4 plus 3, 7 minus 1, 6. So again what we will get? We will get 6 carbon atoms. So 6, 8, 10, 12, like that the chain goes on. And the chain will go on unless and until we get the desired number of fatty acids. Right? So we can say that again same cycle to get desired number of carbon atoms in the fatty acid. Okay, so we can say that if we have to synthesize one molecule of palmitic acid, 8 acetyl coenzyme A are used. So 8 acetyl coenzyme A, right, plus 7 ATP plus 14 NADPH, right, they will form one molecule of palmitic acid, palmitate, plus you can say 7 ADP plus inorganic phosphate or 14 NADP plus H positive. And if you remember, I mean, like when we discussed about the beta oxidation of fatty acids, same it was there, that one molecule of palmitic acid, okay, undergoes oxidation or undergoes breakdown to release 8 molecules of acetyl-CoA. So in terms of acetyl coenzyme में it remains the same. बाकी if we talk about again the energetics, वहाँ पे FADH2 is produced, वहाँ पे NADH is there, here NADPH is there. But yes, one molecule of palmitic acid undergoes 7 cycles of lipogenesis and requires 8 acetyl coenzyme A to produce. Right? So this is what is lipogenesis, which is occurring in the cytoplasm. Now see, this is, you can say it is produced. Now if we have to synthesize long chain fatty acids, so what happens is elongation, matlab synthesis occurs in the cytoplasm as we saw now, but elongation takes place in the ER. Elongation takes place in the endoplasmic reticulum. And even if we have to synthesize unsaturated fatty acids, we can synthesize unsaturated fatty acids depending upon the enzymes. But again, always keep one thing in mind, कि unsaturated fatty acids can also be produced, but our body does not have enzymes which can put double bond beyond 9th carbon atom. So till 9th carbon atom, if it is there, it will be easily produced. So unsaturated fatty acids can also be produced. Fatty acids can also be produced, but double bond is not incorporated after C9. So before C9 it can be easily produced. That is why we say linoleic acid, linolenic acid, arachidonic acid, docosahexaenoic acid, docosapentanoic acid, they are all essential fatty acids. Why? Because they are all having double bond beyond 9th carbon atom. Okay. So this is lipogenesis. Now coming over to the most important aspect, regulation of lipogenesis. Now see, whenever we talk about the regulation, we always talk about the enzyme. So here the rate limiting enzyme is acetyl coenzyme A carboxylase. Acetyl coenzyme A carboxylase. Right. Now, this acetyl coenzyme A carboxylase occurs in 2 forms again, active and inactive. Right. Now the question is, again we will come over to the 3 main hormones, insulin, glucagon and epinephrine. We know that insulin is an anabolic hormone. So insulin will always promote lipogenesis. Okay. So if we say acetyl coenzyme A carboxylase active, so active form will be accelerated by insulin and inhibited by glucagon. On the other hand, if we talk about inactive form, it will be inhibited by insulin and activated by glucagon. Or we can simply say that in our body, whenever insulin to glucagon ratio is high, whenever in our body insulin to glucagon ratio is high, it will depict what? Acetyl coenzyme A carboxylase will be active. Now, if acetyl coenzyme A is active, just go back in beta oxidation where the rate limiting enzyme was CPT1. So, what happens is when acetyl coenzyme A carboxylase is active, so it will simply produce malonyl coenzyme A, right. So, this is the enzyme. This malonyl coenzyme A is going to inhibit CPT1, which is the rate limiting enzyme of beta oxidation. Very, very important. So, whenever in our body the ratio of insulin to glucagon is high, or whenever there is more insulin, it will always undergo anabolic state, producing more and more of fats. Okay. So, we can say that it is always making acetyl coenzyme A carboxylase active. When it is active, malonyl-CoA will be produced, which will inhibit CPT1. And one more additional thing in this is, because whenever we talk about the enzymes, we need to know the structure of that. So, always remember acetyl coenzyme A carboxylase active form is dephosphorylated. It is dephosphorylated. Very important. On the other hand, inactive form is phosphorylated. And this is again in accordance with this type of structure. So, I can say that this is the structure of active form of acetyl coenzyme A carboxylase, and this is the structure of phosphorylated or inactive form of acetyl coenzyme A carboxylase. And it is a well known fact that insulin, this is very, very important, insulin promote those enzymes which are active in dephosphorylated state. Very important. Insulin is always going to promote those enzymes which are active in their dephosphorylated state. Okay. Then if we talk about the opposite one, ये तो क्या है? When there is high ratio of insulin to glucagon, sometimes what will happen is, if insulin to glucagon ratio decreases, insulin to glucagon ratio decreases, what will happen? That means now acetyl coenzyme A carboxylase has to stop working. Okay, so I can say that acetyl coenzyme A carboxylase will be inactive, will become inactive. And how it will become inactive? Because glucagon will now promote lipolysis. Glucagon will promote the activity of CPT1. Okay, so when glucagon is promoting the activity of CPT1, automatically acetyl coenzyme A carboxylase needs to be inactive, because the body cannot afford now to produce malonyl coenzyme A, right? And obviously, if it is activating this, so what will happen is the beta oxidation will be will start, will be initiated. And that is why we say there is a difference between LPL and HSL. Right? There is one enzyme which I told you, it is lipoprotein lipase. And there is one parallel enzyme which is hormone sensitive lipase. The basic difference between these two is that this lipoprotein lipase is considered to be a anabolic enzyme, very, very important, which you have to understand. It is a anabolic enzyme. Rest of the differences we have already discussed in detail earlier in my previous videos. And hormone sensitive lipase is a catabolic enzyme. Right. So what happens here is insulin always promotes LPL. Very, very important. And insulin inhibits HSL. On the other hand, glucagon inhibits LPL, but glucagon is going to accelerate HSL, and that is what is exactly happening. When glucagon is promoting HSL, that means lipase will be active, and that means breakdown of fatty acid will be there. Okay, whereas insulin is promoting the LPL enzyme, which is leading to the synthesis of TGs where in the adipose tissue. And basically lipoprotein lipase, where it is present, if we talk about the presence of this, it is present in the endothelial blood vessels. And it is clear from the word itself that it is simply taking out TGs from the VLDL and chylomicrons, that's it. But TGs ultimately are degraded by hormone sensitive lipase. Okay. So the function of lipoprotein lipase is just to extract TGs from triglycerides from chylomicrons or VLDL. And VLDL, whereas the actual function is carried forward, and where it is present, it is basically present in the adipose tissue. So what it will do is, it will actually break these TGs into fatty acid and glycerol, like this. This is very important again, right? And this is the function of this HSL. So this is how we actually differentiate between lipogenesis and lipolysis. So let's see what are the basic differences between lipogenesis and lipolysis. First of all, lipogenesis and lipolysis, or most probably commonly we can call it as beta oxidation also. Lipolysis, or we can call it beta oxidation. Now, what are the major differences? The most important one is the site. Now, if we talk about the site of lipogenesis, it is the cytoplasm or the cytosol. Very important. Even though elongation takes place in the endoplasmic reticulum, but yes, the major site is cytoplasm or extramitochondrial. Lipolysis ki site is the mitochondria. Right? Then lipogenesis always occurs in the well-fed state, whereas lipolysis occurs in the fasting stage. Right. Then lipogenesis mein, if we talk about the rate limiting enzyme, the rate limiting enzyme of lipogenesis is acetyl-coenzyme A carboxylase, whereas the rate limiting enzyme of lipolysis is carnitine acetyl-transferase 1. Right? Then fourth important point here is that insulin or the hormonal regulation, if we talk about, insulin promotes. Okay, whereas glucagon inhibits. And if we talk about lipolysis, we have to make a demarcation that glucagon accelerates HSL, whereas insulin inhibits HSL, which is hormone sensitive lipase, right? And then another important point is that acetyl coenzyme A carboxylase is always active in its dephosphorylated form. And insulin always promote those enzymes which are active in their dephosphorylated forms, right? So this is all about lipogenesis, which is also known as de novo, which is also known as line spiral. Why it is known as de novo? Because of one simple reason, that it is synthesized, the precursor of fatty acid is simple, you can say acetyl coenzyme A, which has 2 carbon atoms. And from a 2 carbon atom species, we are generating, say for example, palmitic acid, 16 carbon atoms. And here a very important example is of fatty acid synthase complex, which is an ideal example of multi-enzyme complex. Why it is an example of multi-enzyme complex? Because it can never work alone. It has a saucepan type of structure. The middle part, it is basically having what? It is having 2 active sites, which is having cysteine SH and pan SH. And 6 enzyme units are there. And these 6 enzyme units are, one is condensing units, reducing unit, and last one is releasing unit. Okay. So if we talk about the acyl carrier protein or the ACP, it has 2 active sites. One is having cysteine SH, the other is having pan SH. Cysteine SH will always accept acetyl coenzyme A, whereas pan SH will always accept malonyl-CoA. Right. So by the cumulative effect of, you can say, condensing unit and this thing, means one cycle will lead to increase in 2 carbon atoms. So it will go like this: 2, 4, 6, 8, 10, unless and until we get the desired number of carbon atoms in a fatty acid. So I gave you one example of palmitic acid because we've been studying the energetics also of palmitic acid, which has 16 carbon atoms. So 8 molecule of acetyl coenzyme A, 14 NADPH will be required to produce 1 molecule of palmitic acid, and reverse is there in beta oxidation, that 1 molecule of palmitic acid undergoes 7 cycles to produce 8 acetyl coenzyme A, right? And always remember, the synthesis of fats always takes place in the well-fed state. That is why it is always promoted by insulin. And lipolysis always takes place in the fasting stage. That is why glucagon always promotes it. HSL ka isme I am just giving you the example. Otherwise in diabetes and all, HSL automatically increases because there the some other fundi is there because GLUT4 transporters are reduced. So that is something else. But in general, insulin will always inhibit this hormone sensitive lipase, right? So this is all about the synthesis of fatty acids. So I hope this is clear to all of you, and this is a very, very important topic because if you can understand this, this and lipolysis are actually reciprocally regulated also, because one is occurring in the well-fed state, other is occurring in the fasting stage, right?